Next talk will be by Dr. Subhra Chakravarti. She is the former director of the National Institute of Plant Genome Research, NIPGR. She holds a PhD in plant molecular biology from JNU and has a distinguished career in plant genomics, including serving as a research scientist at JNU, a visiting scientist at Yale. And her work also includes germ plasm characterization, pathogen host interactions, and understanding the protein network involved in plant stress responses. She is implementing various national and international projects focused on enhancing crop resilience for sustainable agriculture. Professor Chakrabarti also mentors graduate students and researchers, shaping the next generation of scientists in plant genomics.
Okay, so I hope students could hear me before also as well. So I'm going to talk to you about the bird's eye view towards One Health, and I'm going to connect how can we use the multi-omics data. All of you must be hearing about it, and that landscape. And you know, all of our, whether we are human or plant or animal, all of our cells in our body or in a plant system has organelles. So these organelles does different kind of function, and connecting all those organeller function in a crosstalk can actually lead to a very nice novel hypothesis which you can actually understand, and then can understand the disease biology or nutrition biology or whatever you would like to work on in biological system. So because it's going to be a workshop with the students, so I have put a little bit of some of the slides, initial slides, which talk about what is One Health, what are the important diseases, and so on and so forth. And then I will come at the end about what we have been doing on that. So as you have heard about Professor Swaminathan, you know, this pathogen stress accounts for almost 70% crop loss in the agriculture field worldwide every year. So as the infectious disease, you know, the data that comes from WHO shows that it accounted for more than 36 million deaths in human in the year 2021. When you try to see what is One Health, there are several fungal pathogens, including Fusarium that I would be deliberating later on, have the ability to infect not only the plant or the animal, but also human. So that means they are cross-kingdom pathogens. So that is where the One Health comes in a very big way. So this is one area which is very, very interesting to us. At the same time, as you have heard from Professor Swaminathan, the other angle that is so very important, that is the nutrition and the nutrition security. So little bit about where do we stand today. 13% of world population suffers from nutrition insecurity, and then malnutrition is accounting for 35% of all child death, 11% of global disease burden. The other side of the coin is overnutrition, where we are seeing the change in the food habit, particularly for younger generation that we have been seeing. Earlier it was more of a Western food habit, but now day by day we are also seeing that in India and in the Asian countries, the younger generation are more going to those kind of the food habit, which is little bit of a concern, because that might lead to a situation tomorrow in our own country where it is now that 40% of Oceania's population is obese. So where do we stand in India? Because I'll not be talking about nutrition work, so I'm just putting it that India is where here, and that is where we come within that threat of the second level of threat after African countries. So nutrition is very, very important, that we need to think about where soil health is so very important, and also some other aspects of that. So let's go back to the climate change. You know, this work has come from a global burden of disease that has been told by the Institute for Health Matrices and Evolution. It's a little bit of a older data, but what it tells us that the global ranking of the risk factor, that if you see by total number of death from all cause of all age, across sex in 2017, that shows that dietary risk factor topped the list. And this diet is so important that we eat 70% of human diet comes from the plant derived food, whereas 30% comes from the animal derived food as per the FAO prediction, FAO calculation. And therefore you see there are infectious diseases, there are, you know, what we call as non-infection disease. All of these are very important for the ranking of risk factor for the human death. If we try to see how this is actually these YLDs goes from the day a child is born, from the day that we are lived. So the life expectancy has increased, and now people are living more than 90 years, and how it impacts. So I would like to draw your attention in this area, which is very much where we have been growing, and that's the child to the adulthood. And that's very, very important that we should get proper nutrition at that time, because all our cognitive ability is there from the at least somewhere around 5 years, 7 years up to the 12 years. And at that time, if you see, what is very important, that we have all those kind of deficiencies which leads to many kind of entities, malaria, enteric infection. But not only that, these are the two very major important issue, which is maternal and neonatal condition and nutritional deficiency in the child. Mortality and in the child, if not mortality but the morbidity below 5, in India we have real concern, not only in the world but possibly in India we have more concern than rest of the part of the world. And then if you see here, you would find then as we grow old, and then it's a change in the dynamics of the diseases, and those are more of NCDs and diabetes and CKD and many other diseases, that also where food plays an important role. So now we are talking about One Health. Now this paper came in Science a few years back by Fisher, where they showed that deaths attributable by AMR, which she talked about, Professor Swaminathan, that is antimicrobial resistance, how that's going to impact the world by 2050. So if you try to see, AMR would be the major cause which will take almost 10 million lives on earth by 2050. That is a prediction. And then if you see, that actually goes beyond the death comes from cancer or any kind of other disease, diabetes or infectious diseases. Where do we stand as of Indian situation? You see, we are in a very, very critical stage to actually think about, from now where we are standing 2025, we have 25 years in our hand, how come you can mitigate this kind of a problem, which would be a real threat and comes from the AMR. So what is One Health? You know, this One Health is not a new concept. It started in 2004 by, WHO and FAO and so other important organizations, where in 2004, the One Health concept talked about linking the human health, animal health, and the ecosystem health. But many years after, people realized that plant also is part of our ecosystem. And in 2021, 4 organization panel came up with FAO, WHO, and UNEP. And then they inducted within the One Health from a tripartite to a quadripartite high-level expert panel, and they included plant into the One Health concept. So therefore, we know today that in the human health, 60% of human disease have their origin in animal. We also know that in animal health, the disease in food producing animal globally account for loss of almost 20% of the production that we see. And when we come to the plant and the ecosystem health, that any emerging disease in the last 30 to 40 years resulted from the encroachment into the wild land and change in the demography that has been shown in the world literature. So now for One Health, we talk about quadripartite problem. Now, I would be deliberating on a topic which is a multi-host resistance. So what do we mean by this multi-host resistance? Which caused by cross-kingdom microbes. As I showed you in my first slide, that there are many diseases which can happen because of one particular microbe that that has the ability to infect across kingdom. So I'm putting in this slide some of those pathogens. For example, agrobacterium, which can have in a human a bacteremia, but in plant it causes a tumor that we call as crown gall. Like for Burkholderia, we know cystic fibrosis in human, we also know pink rot in onion. Pseudomonas aeruginosa, cystic fibrosis, again in human, but it causes in onion and a disease and wisteria. So as in case of Staphylococcus, there are many other human-related diseases, but it also causes Arabidopsis, root infection and death. Alternaria is very known to have impact on our respiratory problem, but it also does wood rot. Similarly, penicillium, verticillium, and where we have been interested in our lab is fusarium, which is causing human in an immunocompromised patient the immunostimulatory or immunosuppressive effect, but in plant it has a huge impediment on a disease called vascular wilt disease. So therefore, this is a very, very interesting area to deliberate on and to understand how different hosts actually respond to the same pathogen. So, we work on fungal disease, which is fusarium. And why fungal disease? Because if you see that plant infecting fungi are actually increasing. There is a database which is ProMed. This is a little old data, sorry about it, but the thing is that the story remains the same. This has a battery, or it's not actually. I can pick up this laser, right? Okay, fine, it will work. So if you see that as the animal infecting fungi are growing worldwide, so as the plant infecting fungi. What is important being in India is that we have more of plant infecting fungal diseases in our agricultural field rather than in the clinic, but which is a very, very different scenario if you think about the Western world, particularly the US. So there, the animal infecting fungal diseases or human fungal diseases are more in contrast to the Indian agricultural system. So now, what do we know about the fungi? There are more than 1.5 million species on fungi on the planet. However, it's only 10,000 of them can make infection. So as I said that there are almost 70% crop loss worldwide in agriculture because of the infectious diseases, but out of those 20% comes from crop destroying fungi. Now, out of those, there was a paper came from Royal Society publication quite a few years back, where Caroline Dean's lab showed that 5 of the fungus, Magnoportia, Botrytis, Puccinia, Fusarium, are the top 5 phytopathogen. Although it has been said in that Royal Society publication in 2012, but still today they remain the same 5 topmost fungal phytopathogen. We don't forget about how fungi can infect an animal, which was frog, in kind of over 30 years, a cytrid fungus actually destroyed 200 species of frog in North America in 2007. So it has been said that the global mortality due to the fungal infection would exceed malaria or breast cancer and will be comparable to tuberculosis and HIV. And that has been said in another Royal Society publication a few years before. And I have shown you how many of these fungi can lead to ability to infect both plant and animal, which had been talked by a UC Berkeley scientist very renowned in the plant pathogen field, Brian Statkowski. In 2001 he predicted, but today we know it is the real situation. So with that, why AMR is important in fungal species reported globally? If you see that this data came from a science paper where they have shown, they studied the whole fungal infection and global antifungal resistance from almost 1977 to 2017. And if you could follow that in the plant, in agriculture field as well as in the animal, because of this antifungal resistance, we have a huge problem of AMR. Why this happens? That is because there is a parallel driver of fungicide resistant both in the field and the clinic. Why so? If you go back to the stress to our medicine that you take for fungal disease, for example, nystatin, as well as if you think about the fungicide that the farmers are spraying in their field, so in both of those, the main molecule is the azole derivative. And this azole derivative for this nystatin came in place after 1950s. However, if you think about the agriculture, before 200 years, people used those fungicides. Now, the major challenge to combat AMR, can we find out a molecule which is not the azole derivative? Because if you keep on using the same molecule to mitigate a problem, that creates antimicrobial resistance, because there is a term called coevolution. So as we evolve, our machinery in our immune system evolve, or in the plant immune system evolve, there is a coevolution of the pathogen as well. So pathogens are also very clever organisms, although they are maybe prokaryote or very lower eukaryote, but they also cleverly change their arsenal. And because of this coevolution, we need to change our combating molecules, and therefore it's a huge challenge today. Can in agriculture or in clinic, one can come out and make other molecules which is not a azole derivative, so that you can come out, one aspect would be to come out of the AMR. Now, therefore, in our lab, now I am trying to go to what we have been doing. We are interested to work on a plant animal human health connecting multi-host resistance. That is what I would be talking to you today. We have been working on protein nutrition, which is so very important. I don't have time. Maybe next time I would deliberate on that on a huge scale, where we are doing a mission program now to come up with rice variety with more of the protein and essential amino acids, and how we do, and we have worked on anti-nutrient management earlier, where anti-nutrient like oxalic acid is a health concern for human. If we eat more of oxalate rich food, that leads to kidney stone and many other diseases. And how we do, we ask our question by developing networks using system science, and where we integrate multi-omic data, we develop our hypothesis, find out the hub protein or the hub molecule, and then try to interrogate them, try to understand their function by using molecular biology tools, and then if something comes up, can we relate that or do application work. So that is what we have been doing. Now, I talked to you that every organelle has different function. If you look into it, this is a plant data, it will be the same in case of the animal or human. Outermost layer in plant is a cell wall, which is not there in animal or human. That is the one exception. And beneath that we have the cell membrane, we have the cytoplasm, we have nucleus, we have mitochondria, which is a powerhouse. In plant, another exception, we have chloroplast, we have vacuoles everywhere. So what happens when there is a threat, whether it is coming out of the stress response or a nutrient response, that is perceived by many of the molecules that we call them as receptor, which are there on the outer surface of the cell membrane or the cell wall. So they perceive the signal, then there is a signal transduction that goes through this cytoplasm by many interaction, and then ultimately this whole signal goes to the nucleus, which we call as the the regulatory hub, and within the nucleus all the transcription that happens, and that makes all the RNA molecule. Protein gets synthesized, coming back also in the ribosome, and then you see many of the molecules from where all the primary metabolites and the secondary metabolites, whether irrespective it is plant, animal or human, get synthesized, and that actually impact our whole metabolism under a stress condition, under a disease condition, both in plant and animal and human. So, we have been working on the emerging role of organelles in this kind of cellular processes, and using all these whole cell membrane, chloroplast, secretome, nucleus, cytoplasm, cell wall, and these are so many papers that come from this study. But very importantly, the next question is, can we translate them? And yes, using this system biology approach, we could actually come up with so many important genes and proteins in rice, chickpea and wheat, that now we have been taking it to develop climate resilient crops in the lab as well as nutrient enriched crops. So, coming to today's discussion point is the fusarium disease. So, what does this disease do? This, if you look into the evolutionary scale, fusarium came in this earth 50 million years ago. But if you see their host, which is a dicot we work on, that is Cicer arietinum, and in that case it came in this earth somewhere around 280 million years ago. Worm, which is we use as an animal model, C. elegans, which came in existence 350 million ago, and human came in this earth almost 180 million years ago. But the small Ascomycetes fungi can actually cause disease in plant, in human, in animal, also in cow. So, we have been interested in the question to answer what is the molecular basis of this multi-host response. Now, to do that, first we need to understand the disease biology. So, if you see this fusarium which makes spores, conidiospores, and this macroconidia and the small ones are microconidia, they actually goes from the soil, it is a soil borne disease. So, it goes through the root to the collar region, and it then goes to the, in the plant, if a little bit of school, maybe you are physical science student, but you can always remember your school biology, you know that in plant the water and the solute goes transported through the xylem vessel. Now, this xylem vessel gets choked by the fungal mycelia, and what is the impact on that? Because of this choking in the xylem vessel, you see a drooping, and that is why this disease is named as wilt disease, because the whole plant wilts and ultimately it succumbs to the death. Now, what happens to the fusarium? In the fusarium, if you see, so you grow normally the fusarium, it grows, not fusarium, sorry, C. elegans, the worm grows on OP50. So, its natural food is a bacteria, but we ask the question, can it grow on also a fusarium as its food, because fusarium can also give a carbon and nitrogen source for its growth. So, if you do that, what happens? This is the slide where you see in 24 hours both the OP50 grown worm as well as the fusarium grown worm are still alive. But what happens in 96 hours? The OP50 worm is still alive, but then the fusarium completely dead. And what happens? It dissolute the intestine of the worm. So, there is a parallel. If you carefully look from where the solute and the food is going, both in the plant xylem and in case of the animal, that is the intestine, it gets actually affected by this particular pathogen. So, next what we did, as I said, we used integrated omics approach, that is where we build our network, and we have been working on the genome variation, epigenome variation by high throughput sequencing. We also have been doing transcriptome by NGS sequencing. We then go to the proteome, phosphoproteome acetylome for the modifications, enabled changes in the proteome, and ultimately the metabolome. And we use lot of high end technology based on the mass spec and the GCMS, and then we actually integrate them into a system biology approach, and then this data integration with computational tool, which possibly Shamal's group is also working in some other angle, and then we find out the genes which are important on the protein, we go for functional analysis. So I would be now showing two such example, one on the plant, another on the animal. So if you try to see, in our RNA sequencing data, we challenged chickpea, which is the third most important food legume worldwide. India is the largest producer, highest producer of chickpea, but still we import chickpea from Canada and Australia. Reason because whatever we produce, that's not sufficient for our population, and one of the major cause of the less production in chickpea in India, the topmost disease is the fusarium wilt disease, which globally goes to the 10% of chickpea production decline per year, not only in India but worldwide. So we initially set up a whole system of infection process in the lab, and then we challenged the chickpea in the seedling stage by infecting the fusarium, and then we went for a time course analysis from very early time point, from 6 hours up to the death, that is 5 days or maybe 10 days, and collected the tissue, and using those tissue we have done all this system means multi-omics data. Now what we have learned from the transcriptional landscape, in chickpea we have 8 chromosomes. So the majority of the deregulated transcripts actually came on the chromosome 1, and we wanted to understand, I told you that nucleus is the regulatory hub. So we wanted to know what are the transcription factors which actually regulate this process, and in our data what we found that there are 5 transcription factor family, BHLH, MIB, ERF, VRK and GATA, which showed the top most hub proteins into this transcriptome data. So when we wanted to understand the Arabidopsis data, which is a guinea pig of plant biology, and in that data already another group published, we also actually reanalyzed that data, and it showed that in case of Arabidopsis also BHLH topped the list. So that tells us, suggests us that both in the plant, whether it is a chickpea or Arabidopsis, BHLH and MIB are 2 topmost transcription factor family which actually regulate this whole process. What happens, then the next question, what happens in case of the C. elegans? In case, yes, C. elegans. So in C. elegans we have 6 chromosome, and if you see the deregulation is mostly onto the chromosome 2 and chromosome 3. However, surprisingly, in the animal, it is not the BHLH or the MIB transcription factor, but it is actually the homeodomain, ZF and hormone receptor family. So we see whether it is the same pathogen, even if it is the same pathogen, in the plant and in the animal, there is a diversion of the transcription factor family which actually mitigate the whole problem. So that is the first interesting point that we understood. Then we wanted to understand the co-expression network analysis, where you could see that we divided modules, and this will be going to the next slide. So you need to connect this slide with the next slide, where I would ask you to put more focus onto this blue box, where this blue box containing, what is blue box, I will come to my next slide, and these are one class of molecules which we found both in case of the chickpea as well as in the worm, followed by this cyan and this cyan molecules. These are the 2 major regulatory hub that came out. That is where the similarity is also there, that which kind of biological processes getting actually impacted by this particular pathogen, that remains the same, but the players into those pathways are different. Now in this case we found positive correlation with many of these molecules, so as for the negative correlation in both the case. Now what are this cyan and the blue? So this is the blue which shows the proteins or the genes which are related to wall remodeling, signaling, protein turnover and metabolism, and the same thing we see in the worm data is also signaling and disease progression. So, in the plant data we find four major regulatory hubs. In case of the worm, it is two major regulatory hubs, but two of those, blue and the cyan, are same. One deals with signaling, another deals with the transcription, translation and DNA replication. However, if you see the major hub proteins, they vary between both the blue hubs as well as the cyan hub. Therefore, the players which within that hub is controlling the whole process of immunity against this fungal pathogen varies in plant and animal. Now, from here we then wanted to develop the proteome data, because if you know DNA, we have a central dogma where DNA actually makes RNA by the process of transcription, RNA makes the protein by the process of translation. There are a lot of post-translation and modification which regulate the function of each protein, and therefore we analyze phosphoprotein and the acetylation. I do not have this data ready for acetylation, students are working. I will be showing you the data of the phosphorylation, and the ultimate thing is a metabolome, and therefore we study metabolome. So, at this slide I am actually showing you our data on the proteome and the phosphoproteome in chickpea as well as in the worm. So, what you see here, here also you see this transcription translation and transcription translation are the common deregulation that we see both in the plant and the animal. What is different here is the signaling. When we think about the signaling, we find that in the phosphoproteome data in plant, not only the G protein signaling, but the serine threonine kind of kinase is also very important. But in case of the worm, it actually goes by G protein signaling. So, commonality is the G protein signaling. However, plant has other second diverse pathway also to regulate the proteome. Now, from here also then we tried to merge whole data of all these proteoforms and build our network using cytoscape, and what we find in worm, we got somewhere around 238 nodes, and they can be connected with more than 6,000 ages. That means this 238 protein or RNA transcript can interact with so many of these other protein and transcripts. Ages are nothing but these lines that you could see. That means, let us say this protein do not interact only with this other protein, but it can also interact with this protein. So, how many more protein it can interact? Like all of us are sitting here, we work in collaboration, like IMSC and MSSRF is making a collaborative workshop. Similarly, within the cell, so all the proteins and genes, they do not work in isolation, they also like to collaborate and work together to bring out a function. So, from here we actually narrowed down to our all data, and from this all data what we ultimately came on the enrichment analysis that linked the disease responsive nuclear proteoform. Here if you see, the chromatin remodeling is very common in both the wilt disease and the worm disease, where in chromatin we have histone proteins. There are 4 kind of histones. Out of those, histone 2 and we find histone 2 and histone 4 became the more variable, and we have been now developing the acetylation and whole histone profiling on both C. elegans and plant as well. And other common thing that came up was basically the kind of cell cycle regulation in both the case, and we have common and divergent players in the cell cycle regulation. And then we also have DNA replication, which became common, but if you see mechanosensing, because it is worm, mechanosensing is a major response of worm in animal. So, you see those kind of proteins are getting highlighted, whereas here we do not have those kind of protein. So, from this data then we went ahead and tried to integrate all the genome data, transcriptome data, proteome data, phosphoproteome data and metabolite data. So, if you follow this circle, the outermost this block represent what is happening in the genome level. Within this, the ash gray color represent the transcriptome changes. Within this, the red color is the proteome change, and the green color is actually basically the the metabolite changes that is happening. So in that case, what we see that how different pathways are changing both in the fusarium, chickpea and the worm. Now from here we then came up summarizing this pathway enrichment, and what we could found, we found common pathway between the chickpea and worm. We also found the enriched pathway in the chickpea and enriched pathway in the worm. So you see some of the signaling are very different in worm, which is AMP signaling, chemosensory regulation, intestinal patterning, antiporter cycle, neuronal pathway, because in plant we don't have all this, but we have some certain other molecules which does that job, right. So in that we basically came up to this with the divergence and commonality between a fungal disease where the fungi is the same, host are two different hosts across the kingdom. So from here, my last 2 or 3 slides would talk to you about one of those molecules, although we have been working on more than one molecule to functionally characterize them and try to see how immunity develops against this pathogen both in the plant and the worm, but I'll take only one example. So let's start with one of the example what is happening in the plant. So in that case, we found out and termed from our data IRF, immune responsive factor. So in this case we found IRF1. Question was whether it is a major regulator. Let me tell you, this is one of those transcription factor which belongs to the BHLH family of protein, which was a topmost hub protein. In case my first slide that if you, one of the first slide that you remember when I started showing you the transcriptome remodeling. So from here we found out there are more than 180 BHLH protein in chickpea, but in our data it was 6 of them lighted up, and we were interested in characterizing IRF1. We found out that in fungi infection it is upregulated. At the same time, there are PAMs, which are pathogen associated molecular pattern. Chitin is one such PAM in case of any fungi. So we asked whether this is also chitin responsive. We found that yes, indeed, it also is chitin responsive. We actually tried to localize them, and as you could see, this protein is localized in the nucleus being transcription factor. That was our expectation. And then we did to find out their interacting partners that I said that each protein works in collaboration. From our 2 hybrid data, we found out many of the collaboration. Next question was, what are the downstream gene that it regulates basically. So in that case, what happened, that what we found from our data is that it regulates some other gene, and which turned out to be a protein which can be related to cell wall component. So the cell wall in the plant is a mesh of carbohydrate and the protein. So it targets one of those carbohydrate molecule in the cell wall, and it actually make its synthesis down. So therefore, that is how it basically regulates the immunity. So coming to the one example of the worm, what happens in worm, you have more than 300 neurons. There are 2 class of neurons, ASC and ASI. What in our data we found that because of time constraint, this is a published data, anybody interested can go through the paper. So what we found is that in the neuron, 2 of the major regulator we found as DAF16, which is also a forkhead transcription factor, which was the first topmost hit, and DAF16 is a very, very well characterized in the worm for its development, but nobody characterized it for the fungal infection. So in our data, first time we showed that DAF16 also is very important transcription factor for the worm immunity. And along with that, we found SAR2.1, which is a histone deacetylase. So histone pathway is also very important then in case of the worm. So we did a lot of mutant analysis by using RNAi. You know, the RNAi was discovered in C. elegans, and that got Nobel Prize many years back to Andrew Fire and Craig Mello, and that RNAi is a so nice and easy system to understand any biology, because you can do very easily the double knockout, because the knockouts are there and then you do the RNAi of the gene that you are interested, you get double mutant and you can do it. So to summarize the data, what we found from our double mutant analysis, that in worm there are 5 immune pathway. So DAF16 actually regulates the insulin IGF-1 pathway and not the other pathway, and in other pathway that works with DBL1 pathway as epistatic pathway. So from here, the next question was, there are 6 isoform of DAF16. Again, I take you back to the different localization in different organelle, in this case organ. So in DAF16, 6 of the orthologs are there, out of which we found in our data the intestinal DAF16 is actually doing the job, not the epidermal DAF16. In case of the bacterial infection, it has been shown that the epidermal DAF16 is the major player. The difference between a Pseudomonas, which is a bacterial infection in C. elegans, versus a fungal infection, Fusarium, it remains the DAF16, but different organ DAF16 actually is taking care of the immunity. So what you see here, we then took a transgenic worm of DAF16 tagged with GFP, and if you see in the control where there is no pathogen stress, DAF16 is there in the cytoplasm. Moment you give a pathogen infection, DAF16 travels from the cytoplasm to the nucleus, and nuclear localization of the intestinal DAF16 actually is important for giving the immunity. We came up with this model later on, that how it happens. Basically in the ASI there are GCY5, GCY4 which are transmembrane receptors, and that actually goes and starts the signaling. It goes to a non-neuronal nuclear DAF16 which is intestinal DAF16, and by the use of CLEC, ILIS and SPP, that actually develop the immunity against this Fusarium in the worm. So with that we came up with the model, and in this model we found how this whole process starts from the cell membrane, goes to the cytoplasm nucleus, and through the PCD gives the immunity to both the plant and the worm. To summarize whatever I have discussed with you, we did a longitudinal spatiotemporal multiomics work to derive the biomolecular network, and then what is important is the 7 transmembrane receptor LRR and serpentine receptor was found to regulate this immunity in the worm, but in case of the plant this whole signaling is very different and it goes through a folate and glutamate receptor in case of the plant. And the other important thing that we understood is in plant it is the oligosaccharide signaling, whereas in case of in the worm it is more of a G protein pattern sensory neuronal pathway that affect the immunity against the Fusarium. And finally we came up with this IRF1 as a major regulator of plant immunity against Fusarium for chickpea, whereas DAF16 played a major role in case of the worm. So with that I would leave you with some questions that in the lab right now the students have been working, that what are the functional effectors. Effectors are those proteins which are a pathogen protein, in this case the Fusarium protein, which can make the disease, and there can be PAMP, there can be also effectors. So we have been developing effectoromic study against Fusarium. Then what are the other immune pathway that operate both in human, that is what now we have been collaborating with NIH and try to figure out using macrophage system, and what are the other organelle crosstalk, and what are the common and unique epigenetic marks. So this epigenetic mark work is going on in the lab. Now I'll leave you with a One Health session with some key thoughts. So the thoughts would be, how do we converge the human, animal and plant health and the health of the environment? What is meant by precision agriculture? How that can be impacted? How can the study of plant health research impact onto precision agriculture and medicine? In what ways does multiomics and data science research can tangibly contribute to this One Health concept? And what is very important, what is the role the industry can support our research in the One Health, either for the crop or the farm animal or the food or the bakery or brewery or whatsoever food we intake actually. Can there be a newer and more specialized method or approach that can be required to support this development of the One Health research globally? So with that, I come to the last slide, and this is the most important slide of all my slides, where you could see people who actually contributed are these people, some of my postdoc and some of my PhD student who started the lab. Nashiman actually started this work, who is now a faculty in IIIM Jammu, and then it was followed by Archana and Pooja, and now Shobha is working on epigenetic part. Konika is a postdoc, and Rajul is also a research fellow in the lab. Rajul works on all this integration work on computation. So with that, my funding came from basically SCRB and also my own institute and DBT program. And currently, as I said, for the human part, we have now entered into a collaboration with National Institute of Allergy and Infectious Disease in Washington. So with that, thank you all, and I would be happy to take any question.
Yeah. Any comments? Remarks? Yeah. Sure.
So there was a slide which was comparing the pathways between the worm and the chickpea. Were you able to find any analogous pathways? Like you told this neuronal signaling won't be in worm. So, sorry, it is in worm but won't be in the plant. Did you find any analogous
Yeah,
pathways?
I showed you that, if you remember, that there are analogous pathways, which is particularly that we have been working on. It's on the particularly on epigenetics, that is the histone pathway, and there the histone 2A and histone 4 are the common entity between these two. So the regulatory pathway, there are similarity. In signaling, if you see, G protein is also common between these two, but there are divergence.
Here, please. And okay.
Very nice talk. And this comparison between plant and worm is very enlightening. But when you talk about the changes that are going on, in the case of worm, there are mechanosensory pathways that are different. So I wonder whether it contributes to DOR state, some kind of a dormant state of the cell again.
So very nice question. So yes, it goes. So in the worm, it basically two of the things that you observe. One, DOR state is not there in fusarium, but there is an avoidance stage. So if you see, there are some of the genes which work in this pathways, avoidance related pathway. That we have already published in this paper, but DOR stage doesn't happen.
Right. Okay. Okay.
Yeah, that's it.
Thank you, ma'am. I have one question that maybe I missed that. In both of the cases, whether you have used the same species or the different species like Oxysporum.
Very important question. So my first slide had one killing graph, if you have noticed that, where I showed you the infection process. Yes. So if that killing graph you see, fusarium, there are different species, you are very correct. So we work in chickpea on fusarium oxysporum, and then it is not only oxysporum, after that, because it is a genotype dependent infection, you have former species. Yes. So below the species level. So in that former species, we have Caesari that infects the legume. We have also the species which can infect Solani, which infect the tomato. Now, in literature, there was many years before, maybe 10-15 years before, one paper came out where they have shown this Fusarium oxysporum former species Solani is also human pathogen. In Arabidopsis, it is not a Fusarium oxysporum, it is a Fusarium some other species. So we first, before setting the lab, we actually got all these Fusarium different species, we infected them with the worm, and we studied which can give the maximum killing. So to our surprise, but we were lucky to see that it is the same species, subspecies, that is a Fusarium oxysporum former species Caesari, gave the maximum killing, and therefore it is the most virulent, and we stick to that. So therefore the data that I have shown to you, it comes from this, not only the same species, but at the subspecies level.
Okay, so ma'am, you have used Caesari only in worm itself.
Yeah, so all the multiomics data is with Caesari and the, as a species, former species Caesari.
And ma'am, does that species is reported to cause disease in human? Like you said, Solani is reported.
Solani is reported. This is not yet reported. This is what I said you
Yes.
that recently. So I am talking to one of the scientists and with NIA, means the National Institute of Allergy and Infectious Disease in NIH, that we are going to see its effect onto the human macrophage.
And ma'am, have you translated this research for breeding purpose?
Yes. So that gene I haven't yet talked to, but I told you that our lab is interested in connecting the, crosstalk between all the organelle. So very recently from a map-based cloning work that we found out a receptor protein, which is on the membrane, but it is a negative regulator. So that means if you actually have more expression of that, that makes the plant more susceptible to this fusarium disease, that is a vascular wilt. So now we have made a CRISPR line, we are making the CRISPR line, but we have used, are you a plant background?
Yes, ma'am.
That's why you are asking me all these questions. So, so in that case, you know Agrobacterium rhizogenes. So this is a root-borne pathogen. So we have already have a data now, not yet published, where by using a CRISPR on a rhizogene, if you knock down that receptor, it actually gives you a better tolerance to fusarium. Now we are preparing that, and it just recently we got a Bill and Melinda Gates Foundation. It's part of that program that we would come up hopefully next 3 years with this line, which will be fusarium, hopefully a fusarium tolerant line.
Thank you very
Yeah.
much, ma'am.
Thank you for the talk, ma'am. It was very interesting. I would like to know that how serious can fusarium be with regard to human health? We all know that it has been very devastating for the plants. How serious can it be for humans?
When I started talking, started my lecture, so I said that in human, uh, fusarium disease is not one of the topmost infectious disease. No, not that way. But if there are reports where there is an immunocompromisation, particularly in eye, so in Madurai there was one lab who worked for many long years with the Eye Institute, and they showed that in eye, fusarium infection is a very important infection. At the same time, if you have a surgery case, and there are literature in Lancet and many others, which shows that it can be a lung infection as a secondary infection following the operation. So wherever, and then also another important part is keratitis as well as a skin based infection from where it affects the epidermis. So these are few that has been shown. Therefore, although it may not be a very topmost pathogen, but we started with the chickpea, and it is the first topmost pathogen. But then when we were working, the students working, then we little by little got interested into multi-host, and therefore we have been working with that because there are incidences in human as well.